Okay, thank you very much. So my name is Thomas Arskog. I'm a BD manager at Xintela, replacing our CEO, Eva Lundgren Åkerlund, today. So we're a Swedish biomedical company located here at Medicon Village, actually, a great place to be. We're a team of 25 people today, approximately, and we've been listed at Nasdaq First North in Stockholm since March 2016. So we develop products in two main areas. So that's cell therapy and in oncology, where we focus on indications with a high unmet medical need. So our current focus in cell therapy is on the degenerative joint disease, osteoarthritis, and in oncology, the brain tumor glioblastoma. And although these are two very different indications, we actually base our product development on the same proprietary integrin marker technology platform. So the marker that we focus most of our work on is an integrin called alpha-10 beta-1 that initially was discovered as a specific marker on chondrocytes or cartilage cells. Later research showed that certain mesenchymal stem cells also express our integrin marker, and that it is strongly upregulated in certain cancer cells, and this is what we utilize in our product development. So over the years, Xintela has developed antibodies that bind to the integrin marker that we use in our product development, and we use them in two main ways. So in cell therapy, we use them to identify and select high-quality therapeutic stem cells, or mesenchymal stem cells, MSCs. And in oncology, we use them to target aggressive cancer cells with a drug. So in cell therapy, our first focus is on osteoarthritis, which is a degenerative joint disease with a degradation of cartilage in the joint over time. It's a huge problem affecting over 600 million people globally, and it's a problem that increases with age. And the global market for OA drugs is huge, and it's growing rapidly. And of course, we want to take a part of that. So we develop what we call an off-the-shelf mesenchymal stem cell product for the treatment of OA, both in humans and in animals. So we use what we call allogeneic or donated stem cells from adipose tissue. So with that, you can use stem cells from one donor to treat many patients. So it's a very cost-effective approach. And we're also aiming for a simple one-step procedure where the stem cells will be injected directly into the OA joint, so you won't need any surgery of any sort. So what makes us unique? Of course, we have competitors out there developing stem cell products for OA and other indications. What makes us unique? So this is explained here on this slide. When you isolate stem cells from adipose tissue or bone marrow or other sources, what you really get is a heterogeneous mixture of cells. So in this mixture, you have some true stem cells, but you also have progenitors and other cell types. And this preparation is what most other companies are using in the product development, and that causes a lot of issues, not the least from a regulatory point of view, because of problems to show product identity and consistency. So we solve this problem by selecting high-quality stem cells based on our marker to obtain a homogeneous stem cell population that we then use in our product development. So with this, we get a homogeneous and consistent stem cell preparation. And that brings a set of advantages, for example, functional advantages. We know that our selected stem cells, for example, have a much higher chondrogenic differentiation capacity, so they have a high capacity to differentiate into chondrocytes, as shown here in a pellet mouse assay. So these are selected stem cells. So shown here by staining collagen type 2, which is found in high-quality cartilage. So in addition, we know that our selected stem cells are immunomodulatory, anti-inflammatory, and that they also have an increased ability to adhere or home to damaged cartilage. And this is really important. So they can find the damage and to the place where they should exert their function. We also have several regulatory advantages with our approach. So as I said, we get homogeneous cell preparations. We get consistent quality between donors and batches. we can use our marker technology as an in-process control and quality control, and to predict the potency of cells, which hopefully will show on the clinical outcome. So we get a defined and consistent stem cell product. We have analyzed our stem cells in a post-traumatic OA model in horses, where equine stem cells were isolated from equine adipose tissue, selected with our marker technology and frozen down in vials, shipped to our collaborator at Cornell University, New York, Dr. Lisa Forcier. So they injected our stem cells in one damaged ankle joint, and in the other got a saline solution. And after 6 months, the cartilage, underlying bone, and other tissues were analyzed. This is some results. If you look at the cartilage, so this is cartilage from a non-impacted area, stained here with saphanin OA, saphanin O, showing a really nice cartilage. This is in the control. Here you can see that in the impacted area, no cartilage at all. You can see a lot of fissures. And this is in the treated joint, much better integrity of the cartilage. And this was a quite small study, 7 animals. It was statistically significant between the groups if you looked at cartilage fibrillation, so less cartilage fibrillation in the treated joints. And also a much better result also when it comes to bone sclerosis, which is another important hallmark for OA. So in the group treated with the joint treated with our MSCs had a better sclerosis score and also a thinner subchondral plate. So we reached several important milestones, positive preclinical results. We now have a scale-up production of stem cells in the bioreactor. And importantly, we have built our own GMP facility for production of stem cells for our upcoming clinical trials. And this is, I think, very important because it builds a lot of value in the company, because as a partner, we do not only bring a unique stem cell platform, but also the manufacturing capabilities. We're preparing for our first in-human study. We have a clinical study protocol in place, and we are finalizing negotiations with CRO and study sites. And we recently had a scientific advice meeting with the MPA regarding the clinical study, and with very positive response. So the next milestones, we are waiting for an approval for the GMP facility. We're going to initiate our first in-human study with our first product, XTEM-OA, for treatment of knee OA. Initiate co-development with a partner for OA therapy in animals, and also do more preclinical evaluation for new indications for stem cell treatment in the orthopedic and possibly CNS indications. A few words also on our oncology project. Here we develop antibody-based products for treatment of glioblastoma as the first indication. Glioblastoma, as you know, is the most common and most aggressive form of brain tumor in adults, affecting some 25,000 to 30,000 individuals per year in Europe and the US. And also here we have a substantial global market for glioblastoma treatments. What we develop is an antibody-drug conjugate. It's a first-in-class approach. We see that our integrin target is strongly upregulated in glioblastoma tumors. So the idea here is to target the cancer cells with an ADC that delivers a toxin that kills the cells, the glioblastoma cells. So we have several positive preclinical results. I don't have time to go into the details here, but we have shown that the ADC binds to glioblastoma cells and has a killing effect both in cell studies and in animal model. So we have a therapeutic target with large potential in oncology. Positive preclinical results that were recently published in a distinguished journal called Cancers. We have identified new cancer indications recently to focus on. And we have filed a new patent applications for diagnosis and therapy of aggressive cancers based on our technology. So the near-term milestones here is to identify antibody candidates, to do proof of principle studies in animal models, that's ongoing, and to evaluate and decide on new oncology indications in addition to glioblastoma to focus on, and to identify partners and licensees. What we have also done is that we have spun out our oncology business to a new company called Xintela. So Xintela will focus on cancer therapy, the products, because it's developed so well over the last 2 years. And all the stem cell therapy products will remain in Xintela. So we're looking for partners and investors for both companies. So final slide, why Xintela? We do have a patented market technology platform with large diagnostic and therapeutic potential, stem cell therapy expertise and technology with unique advantages, and importantly, in-house GMP manufacturing facility for the stem cells. We have animal health projects with shorter time to market. We have a first-in-class oncology target for therapeutic product development and extensive IP covering products, methods, and use claims. Thank you very much.